you're taking healthy animals and inducing certain disease. And the problem with disease modeling is that you're not really recreating a full human disease. You're capturing a snapshot of the human disease. And I thought to myself, is the work that I'm doing in animal models ever going to help people like him? And the answer was a very resound, loud no.
Hello and welcome to the 3DCC platform conversations, where we bring the voices all over the world who are working in the domain of healthcare to build the future in the healthcare domain. And I'm Rohini Kanbarkar, I'm pleased to welcome who has joined me for today's conversation, Dr. Charu Chandrasekera, a recognized leader in the toxicology, regulatory science, and new approach methodologies. Her work focuses completely on advancing the human-relevant, non-animal approaches and also translating the innovative science into the regulatory accepted safety frameworks, I would say. Welcome on this platform, ma'am.
Thank you. Thank you for having me.
And so, before starting the whole conversation, can you briefly tell me how was your journey so far in this domain?
The actual field in the domain?
Yeah.
It has been quite incredible, actually. I think over the last 10 years, we've seen a lot of change, not just in Canada, but around the globe with new laws and policies and roadmaps, strategic plans. So around the globe, there's been a lot of change. And it's rather uplifting to see that everyone is working toward the same goal.
Exactly. And what kind of experiences during your work or the early research years that you had led you to question the effectiveness of how the traditional animal models are being used in the biomedical research?
I mean, I've had some personal experiences, and I've spoken about this before. I did cardiovascular research using mouse models, and the turning point came for me when my own father had a heart attack and required quadruple bypass surgery. And I remember sitting by his bedside in that ward after his surgery. I looked around and I thought to myself, is the work that I'm doing in animal models ever going to help people like him? And the answer was a very resound, loud no.
Yes.
And that to me was a very big turning point, because I didn't go into science to help cure heart failure in mice. It was to help humans, and getting to know the dire statistics. 95% of drugs tested to be safe and effective in animals fail in human clinical trials. Those are not facts or statistics that you can simply ignore.
Yes, that's definitely important. And can you explain me what animal-free science means when we talk about the practicality of this and why it matters so much for both humans and also for the animal welfare?
Yes, it is obviously two sides of the same coin, right? It is not simply an animal rights issue. It's also what I have often said is a human rights issue. When you have, and this is one of the examples that I have spoken about before, think about Alzheimer's disease, for example. 99.6% clinical trial failure rate over there. If you just look at the last 10 years, how many trials have, and we don't even understand the molecular basis of Alzheimer's disease yet, right? There are all these hypotheses and you can come up with all of these things. So what are we doing there? And that's where you really have to think about how applicable are these non-animal methods. Of course, we've learned from animal models. I mean, over the past century, most of the textbook knowledge that we have comes from animal studies. But through all of those studies, we've also learned what the limitations are, the colossal limitations. And so I think it's time to actively move away from that to really understand how do you interrogate human biology, how do you understand disease mechanisms or human biology in health and disease, and also when you have adverse events to toxic chemicals and things like that. It is simply not possible to translate. And at the same time, look at some of these assays that we do, 2-year cancer bioassay, and feeding pesticides to dogs for 6 months. Should we be doing things like that in this day and age? It's 2026.
Exactly. There should be alternatives that we should be working more.
Absolutely. I think that I don't think we're doing enough.
Okay.
To me, I think it's still really slow, but I mean,
Okay.
we're making tremendous progress. I would like it to be a little bit faster.
Yeah.
And then we talked about this a little bit in the sessions yesterday too, right? I know where are we going, and the exciting news is that the US EPA just reinstated, just like a few hours ago, essentially reinstated the 2035 deadline, and it's really important to think about it like that, because at the end of the day, even if we don't meet the exact deadline, we have a goal to work toward.
Exactly.
And so we can really put all of our resources, our brains, our thinking caps on, and come up with pragmatic approaches to do that.
That's wonderful, actually. What are your perspectives? Like, what are some of the scientific and ethical limitations in using the animal models that you believe make them less effective for the human biomedical research?
So the use of animals in biomedical research versus toxicity testing, I mean, there are two different model systems, two different approaches, right? In one, you're taking healthy animals and inducing certain disease. And the problem with disease modeling is that you're not really recreating a full human disease. You're capturing a snapshot of the human disease, if at all even relevant to a human disease, right? So think about even diabetes or cardiovascular disease. Think about how humans develop it over decades of time. There are so many different factors, and there are also comorbidities and other things that you cannot effectively model in animals. If you take out, if you remove the pancreas of an animal or destroy it using a chemical or genetically modify a certain thing, that doesn't necessarily, it's only capturing one aspect of the human disease. So it's really not possible to capture the full breadth of the human disease, and also the mechanisms are not the same. And so I think it's a combination of so many different things where the animal biology does not emulate human biology, and it's become really evident. And like think about everything over the last 20, 30, 40 years, the amount of time and money we've spent on looking at some of these diseases. Like I just said, 99.6% drug failure rate for Alzheimer's, 92% failure rate for cancer drugs, and there are problems. And with toxicity testing, you're taking healthy animals and exposing them to these random, let's make them inhale something for 28 days or 90 days and see what happens. Most of the time with animal data, for toxicology, you're looking at death as an endpoint, or just full organ where organ effects or things like that are very sort of superficial in a way. But with new approach methods, with the 21st century vision for toxicology, you are doing more mechanistic. So instead of death as an endpoint, you're looking at mechanisms of action, and you are trying to capture human biology in a particular context. So it is not one method applicable to all case scenarios, but you are asking the questions in a different way. What is the best way to capture human biology and under certain circumstance, and how do we do that? What tools do we use? And you use more than one tool. It's never about here's one animal test that's replaced with one new approach method. It's a combination of methods that you could use to capture the complexity of human biology from molecular level to population level.
I mean, that's wonderful. That shows how important it is to focus on every element rather than just building and answering one question. It's important that we see each and other compartment and how it's working all together.
That's the beauty of that whole thing, right? I think with new approach methods, we have so much creativity that you could do. And, take a look at some of the things that were recently approved by the OECD, the skin sensitization. So this is allergic reactions in response to skin application of chemicals, for repeated exposure to chemicals. For example, it used to be a mouse assay, the local lymph node assay, that has been replaced by 2 to 3 very simple cell-based assays and some computational modeling. It's incredible.
It becomes an alternative.
Right. So you're not looking at the exact same endpoint as in the animal, but you're trying to capture the mechanism of action and mode of action in many ways, and trying to design assays that would capture the biology that goes from a molecule binding to your skin proteins to what happens in the body. And you can apply the same principles to many other toxicological endpoints or many other biological endpoints that you're trying to study.
And when we talk about so much innovations going on, something that is very important is how regulated all these innovations takes place, right? Like the regulatory body also plays a very key important role in this. So what do you think, how does these regulatory bodies and the international consortia that we have and the policymakers which are there works together to modernize these testing frameworks?
This is the key part. I think everyone has to come together. There are many key stakeholders, right? So the technology developers, the end users, right? So technology developers may be different from the end users, right? So you have pharma, chemical companies, and then you have the technology developers and policymakers and regulators also from different parts of the world, because not everyone follows the same thing, right? So in North America, it's mostly an exposure-based risk assessment, whereas in the European Union, it's more, hazard-based. Whereas you know, when you're going from a risk-based to a hazard-based and that type of thing, so there are differences. Different agencies have different expectations. So I think it's really important for everyone to work together and tell the people who are developing the technologies, this is what we want to see from it. Because otherwise, what I am seeing right now in our field is that we have a lot of different technologies and they're not standardized. They're not quote unquote validated. And so you may have a beautiful model that gives you certain that enables you to interrogate certain level of biology, but everyone's doing it differently. And, one regulatory agency may be like, oh, this is good enough. The other one could say, no, it's not. And I think that that's the biggest thing that we need to do. The next big step is to have some qualification criteria. It would be really amazing because right now, even with ICH, ISO, OECD, yes, everyone's sitting around the big tables and talking about it, but it takes too long. It takes too long to get an OECD protocol approved. But I think we can start being more efficient by having individual conversations. So technology developer should be contacting their own regulatory agencies and having those conversations early. Like, what would you like to see from this? Or if I were to get these data out of our technology, would you be amenable to considering that?
So that would bridge the gap between this, the time gap that we have.
What we have a big gap in regulatory acceptance. Look at some of the most amazing, even organ on chip has not yet received that regulatory acceptance we're looking for.
So this is where we need to still work on it.
It's the key. There just really needs to be, and we had a big conversation about this yesterday. In our sessions.
And that is important, definitely. And when we talk about futuristic approach, right? When we talk about what is there in the future that we look up to, what do you think? Like, where do you envision the field of animal-free science and the regulatory testing in the next decade, or maybe in the next upcoming 5 years? Where do you see it?
That's an interesting way. So part of me. Obviously, I'm really excited at the prospects of where things are going. And I think especially when you think about, where are some of the organ on chips, for example, and the validation work that's been happening at the US FDA. And when you look at the new sort of commitments, right, the FDA roadmap, the Modernization Act, the European roadmap that's coming out and things that are happening in other countries. And even, we have a strategy in Canada. When you think about it, you think that in the next 10 years, there's going to be major advances. But at the same time, I look at, okay, what happened in the last 5 years? Did we reach our goals? And part of me is like, no, we really need to bring more people together, and we need to do these sort of go from having these fancy technologies to having them be useful and be integrated into the risk assessment system. So that's what's missing. But I think maybe in the next decade, that's what will happen. So especially when the EPA says something like, okay, we are going to reinstate the 2035 deadline. I'm not sure we could end it all by 2035 at this rate. However, when you have a goal like that, then you work toward that goal. Maybe we do need to accelerate our process. And I really think it's really important to have a deadline in mind.
That gives you
then it gives you that fuel to really work together and do that. So yes, I think the next decade we're going to see immense change. We're going to have the first microphysiological system approved for regulatory risk assessment. It will be incredible to witness all the changes around the world.
More regulations and policies will work together.
It would be. Yes.
And before we end the conversation, what kind of advice do you have for the young researchers, young innovators who are as courageous to get their steps into this, to get into this path of healthcare, especially in the animal-free testing? What kind of advice you have?
This is interesting because that is one of my biggest things. I do want to inspire the next generation of scientists. That's where the future is. I think all of us who have been in the field for a while now, we're doing our very best. But soon enough, there needs to be people to carry the torch forward. Exactly. So if you think about the next 10 years, by the time all these regulatory agencies around the world are ready to evaluate the next pesticide or the drug using organ on chip and AI and all of these other models, you need to have evaluators in these agencies who understand the kind of data that's coming from these new approach methods. So we need to start training those kids now. And my biggest advice to young students is don't be afraid to challenge the status quo.
Okay.
Every time I teach and do that, question. Science is an evidence-based enterprise, not a belief system. You must question. The only way that you're going to improve science is by questioning it, by challenging it, and figuring out what the problems are and coming up with those solutions. And I think the generation that we have now who are ready to embrace new approach methods, they're not afraid. And I see this in my own students. They're fearless and they're not afraid to talk about that, right? Animal models are not the be all end all. We don't have to worship them anymore.
Exactly.
And I think it's time to really, for the next generation, I have a lot of hope. And I have this very heartfelt story this was a couple of years ago when I was getting ready to host the 12th World Congress on Alternatives in Niagara Falls. I received an email from a student in grade 4. And this is what she said. She said, "Dear Dr. Chandrasekera, I am a student in grade 4 and I have to write and present an essay about an inspirational Canadian.
Okay.
And I chose to do that on you."
That's
and
wonderful.
she said, "I love science. I love animals. I don't want to do animal testing.
Yeah.
Tell me how I can become an animal-free scientist just like you."
That's the start for me.
What more can you expect from that? And this is as young as she's 10 years old in grade 4. That was incredibly inspiring. And even my own graduate students who reached out to me saying, this is what we want to do. I'm very hopeful. I think that after a century of people saying, we need animal models and we need to do this and they're the only way, I think we are coming up with another generation who are saying, no. Like, I don't think any of my grad students who've graduated from the Canadian Center for Alternatives would ever touch an animal model.
That's great. We have a great future incoming then. It was wonderful having this conversation, ma'am. Because we have time constraints, I have to end this conversation. But I would have loved to talk more about all of these things with you. We'll definitely catch up
I'm sure we will again. Well, good luck with everything happening in India. I'm really impressed to see the progress happening here.
It's wonderful, actually. And we look forward to connect with you once again in the future.
Sure.
Someday.
Okay,
Thank you so much for being here on the podcast, ma'am.